I'd like to thank the organizers for inviting me to this wonderful meeting. I'm so sorry I couldn't be there in person at the time of this talk. I'm with my family on a holiday in Kauai, where it's a 12-hour difference. So I'm sorry I won't be able to join you for the panel later on. Today, I'm going to be talking about targeting Alzheimer's disease as a heterogeneous innate immune disorder, arguing that Alzheimer's pathology evolved as a protection mechanism in the brain, leading to conservation of the gene mutations that predispose to this disease today. I'd like to begin with my disclosures over the past 5 years of companies in which I consulted or have equity. So it was Alois Alzheimer, of course, who described plaques and tangles and neuroinflammation over 100 years ago. But the real modern era of Alzheimer's began when George Glenner and Colin Masters first described the amyloid plaques being made of the amyloid beta protein. This allowed our group and 2 other groups to isolate in 1987 the gene that makes amyloid beta protein, APP. And then this set the stage for finding mutations in APP, followed by the presenilins, and finally ApoE as a late onset gene. And all 4 of these genes led us to the same conclusion that beta amyloid is the etiological agent in Alzheimer's disease. Now, the problem is that when this was done in mice, it didn't hold up as well. Before I get to that, let me just say that in my lab, we continue to find genes that support the amyloid hypothesis from GWAS, rare mutations, and for example, atoxin-1 that regulate base 1 transcription, ADAM10, loss of function mutations, and alpha secretase, the chaperone GGA3 that controls base 1 degradation, mutations in ACE, and even new mutations in APP outside of the A-beta domain. So genetics continues to support A-beta. The problem is that in the over the last 30 or so years, in studying the amyloid cascade hypothesis, when you put these early onset FAD genes into mice, basically you get a failure to see amyloid inducing tangles. In fact, that's part of the reason why the cascade hypothesis has so many arrows between amyloid and tangles is one of the arguments was the mice don't live long enough. Well, it turns out mice cannot make tangles. And if you use 3D models like those championed by Du-Young Kim and Se-Hun Choi in our group, 3D human neurocell culture models of AD allow you to see A-beta induced tauopathy directly. This is the first 3D model that we published in Nature in 2014, where by expressing FAD mutations in human embryonic stem cell derived neurons, we could see plaques and oligomers and then plaques in the dish within 4 weeks, A-beta fibrils. And if you wait just another week or 2, you see bona fide tangles, paired helical filaments, phospho-tau from endogenous tau. So this is basically amyloid inducing. If you block the amyloid with beta and gamma secretase inhibitors, you don't get the tangles. If you block with GSK3 beta inhibitors, you have amyloid, but you won't get tangles downstream of amyloid. So this is the first paper. And what we realized was that in these 3D models using the embryonic stem cell derived neurons, these are mature neurons that have a 4-repeat, 3-repeat tau ratio similar to adult human brain. Whereas IPS-derived neurons, this won't happen, so it's more difficult to make tangles in IPS-derived neurons in response to amyloid. And of course, in mice, you don't have this ratio, so they don't make tangles either. So this helps explain the discrepancy. With this model now, we can ask in this 3D model, is it A-beta that's really causing the tangles? And this was basically answered by the 3D non-cell autonomous model of AD, championed by Sung-Soo Kwok in Du-Young's lab. And in this case, various mutations were entered into the neurons that increased the A-beta 42:40 ratio. And as a result of that, increased the amounts of A-beta oligomer in the 3D model. What we can see here is that the higher the 42:40 ratio, the more tangles you see from endogenous tau. But the key experiment saying it's A-beta was here. These are naive neurons growing in gel. You have liquid below with A-beta producing neurons making different ratios of 42:40. You have here a filter that's size excluded where A-beta can get through. And as A-beta gets into the gel and you have more 42:40 ratio, oligomers form here. And then from naive neurons, from endogenous tau, you're making bona fide tangles and paired helical filaments. So I think this argues more than anything else. It's not something else due to presenilin mutations or APP mutations or CTFs. A-beta is inducing tangles, it's the oligomers, and they're driven by the 42:40 ratio here in this model. We can also compare this model by by doing a transcriptomic analysis to AD brain. So if you take AD brain transcriptomic data from Mayo, from the Mount Sinai Brain Bank, and you then do a pathway analysis, what you can see is that in the models where we have the highest A beta 42 to 40 ratio, you see that the transcriptomic profiles and the pathways implicated by that most match between AD brain and the 3D model. These are the upregulated pathways here, namely NF-kappa B, the downregulated pathways here. And you see this extremely significant p-values, 10 to the minus 16, 10 to the minus 6, to show a correlation between AD brain transcriptomic profiles and pathway analysis versus the 3D models, with A beta 42 to 40 ratio being the biggest determinant of when you get a match between the 3D model and AD brain. This is unpublished data from Joseph Park and Du-Yan Kim. So what this says is that yes, A beta 42 oligomers can directly induce tauopathy in these 3D models, even though you don't see that in mice, that cell death can go on to induce microglial activation, astrogliosis, neuroinflammation. But of course, you can get a feedback with neuroinflammation causing oxidative stress that causes tangles. And we also know from the mice that amyloid itself, if you accumulate enough, can induce neuroinflammation directly. Now, we know from resilient brains that you can have folks who die in their 80s with no dementia whatsoever, and then to the surprise of the pathologist, you see tons of plaques and tangles in their brain. These are also called mismatched brains. And what was learned from the resilient brain studies that have been done is that if you have lots of plaques and tangles with no dementia, what is usually the case, or almost always the case, is you don't see evidence for neuroinflammation, no astrogliosis, no activated microglia. You don't see the pro-inflammatory cytokine profiles that are consistent with neuroinflammation. So some folks are lucky enough, probably genetically, to not have a massive neuroinflammatory response to the plaques and tangles. They don't get dementia. So it's no surprise that in GWAS studies, you've seen a domination of innate immune microglial genes. Most AD genes come from GWAS, are expressed in microglia or macrophages. The first of these innate immune microglial genes was found by our group in 2008, CD33, Bertram et al. 2008, in a family-based GWAS that was replicated later by Naj et al. In a case-controlled GWAS in 2011. TREM2 came next, NPP5D. These are probably the most studied. But I would say right now, there are probably about 50 AD genes in GWAS that all bring us back to innate immunity and microglia. And this is how it works. This is a modified figure from Beth Stevens, where you have homeostatic microglia that can either be inflammatory, causing excessive synaptic pruning and cell death, or you could have microglia that are basically housekeeping at night while you sleep through the lymphatic system. They're clearing A beta and other debris. And what we now know is that when TREM2 is up, you get more of a phagocytic microglial cell clearing A beta, less neuroinflammation. Whereas when CD33 and NPP5D are up, it's the opposite. You get more pro-inflammatory cytokine release and more of a pro-inflammatory profile of the microglia. So these are opposite effects of CD33 and NPP5D versus TREM2. So we can now introduce this into the 3D model, and this is now a 3D neuroglial triculture model that this is the unpublished model that is now evolved off of the first model we published with Hong Sang Cho in 2018, Nature Neuroscience. This is a much more adaptable model where you have a 96 well dish here, and in each well, you have the 3D model below. So here's Alzheimer neurons and astrocytes making plaques and tangles, then a layer of gel and a human microglia. These are IMGLs from iPS cells, are sitting on top. And what you can see is if you have control neurons in the bottom, microglial cells come in pretty slowly. Whereas if you have neurons making plaques and tangles, like I showed earlier, the AD model, what you can see now is that you see many more microglial cells come in. And here's what happens. So these are the normal neurons, naive neurons, growing in 3D. You see a certain amount of blue microglial cells come in. They're peacefully coexisting. The microglial cells are clearing array debris, doing their phagocytic job, housekeeping job. But in contrast, if you have neurons making plaques and tangles in the dish, many more microglial cells rush in much faster. And you can see in real time here, these activated microglial cells, they're causing astrogliosis, and you can see them directly pruning synapses, causing axotomy, devastating the neurons in this dish. So this is basically neuroinflammation following plaque and tangle formation and recruitment of microglia in one model in a 96 well dish. We can also see that when these activated microglial cells come in, you get a big increase in the amount of phospho-tau and tangle formation. This is probably because of astrogliosis, oxidative stress causing more tangles and thus more cell death. This is all modeled in the dish. Now, some folks are using a model where the IMGLs are instead introduced into transgenic mouse brain, and you can see similar effects of these microglial cells becoming activated. And what's interesting is if we look at transcriptomic profiles of the neurons and astrocytes in the dish model where you add IMGLs versus AD mouse models, it's very, very similar. You see a very similar transcriptomic profile from microglial cells entered into an AD brain in a mouse versus in AD neuropathology in this 3D model. And of course, the 3D model is much easier to use than the mouse, 10 times faster. And for drug screening, which I'll talk about later, 10 times cheaper. So this is the larger pathway pathway that I can show. I know it was complicated, but let's start with number one. You have A beta production. A beta can be exported through the blood-brain barrier and regulated that way. A beta can form oligomers unless the A beta is induced, is phagocytosed by microglial cells here. You can also have enzymatic degradation of A beta by neprilysin and IDE. So many different ways to regulate A beta levels so that you don't get too many oligomers, because these oligomers, as we showed in the dish, directly induce tangles, induce synaptic degradation. As tangles are released and as neurons die, this debris can be taken up by microglial cells and activate them into a pro-inflammatory state. Of course, normally what you'd want is microglial cells to clear A beta and stay more in this phagocytic state. That's exactly what Aduhelm, for example, does, is it induces microglial phagocytosis of A beta. But once these microglial cells are activated, they release pro-inflammatory cytokines and chemokines. This induces astrogliosis. Now you have reactive microglia, reactive astrocytes. This is now causing inflammation, oxidative damage, synaptic pruning, mitochondrial dysfunction, ER stress, ultimately the cell death. So this is the pathway we know about. Now, if we look at these events through the lens of brain imaging studies over the years, we can see that the plaques come very early, up to 30 years before symptoms, and the phospho-tau is following. And it's only as neuroinflammation starts to cause rampant amount of cell death, then you start to see onset of symptoms of cognitive impairment and dementia. So what this tells us, if you put all this together, is that if you take this diagram, everything up here, amyloid deposition, oligomers, induction of tauopathy, really, or trying to get microglial cells to clear more A beta like Aduhelm does, this is all best for prevention, okay? These are early events. And if you want to treat the symptoms, you really have to come down here, optimally prevent neurons from dying or somehow try to maintain neurotransmitter release and sustainment neurotransmitters like Aricept. But the big question is, if you stop neuroinflammation as you come around this corner, will that still be more proximal to prevention, or will stopping neuroinflammation be more proximal to treatment of symptoms? And this experiment hasn't been done yet, but now you're seeing companies do CD33 and TREM2 studies, RIPK. So I think soon we are going to have some evidence for whether blocking neuroinflammation is able to treat symptoms and not afford prevention. So in the future, the way we're going to prevent Alzheimer's disease is early prediction, early detection, early intervention. For early prediction, we have many Alzheimer genes beyond the original 4 now. In fact, from GWAS, we have 94 validated loci, and across these loci, there are 174 candidate AD genes. In some loci, there's just one gene here, like SORT1. If you look here, for this next locus here, you have 3 different genes. So there's still more work to do in GWAS to identify which is the right gene in each of these loci. But that's 94 loci, basically 94 genes we have to pick from 174 candidates moving forward. For early detection, we have brain imaging. We have PET scans, MRI for amyloid, PET for tangles. We have an increasing number of CSF and blood tests that can look at A beta deposition, looking at A beta 42 to 40 ratio in CSF and plasma, phospho-tau levels, neurofilament light chain, and neuroinflammatory markers. And we also have a growing number of digital technologies that can detect changes in the brain. This is one I helped develop with a company I started called React that does eye scanning in the context of a neuropsych exam and language and verbal language analysis at the same time. So we're getting better and better at early detection. I want to now mention, just as a side, just for one side story, that we recently tried to ask, can we find other plasma-based biomarkers? And we hypothesized that perhaps pro-inflammatory cytokines in the periphery, in the blood, might predispose to increased risk for Alzheimer's. Well, we did this with Risa Sperling and the Harvard Aging Brain Study. So these are 300 normal elderly, and they're being tracked for 6 years for cognitive changes. Their blood is being drawn, they're getting neuropsych exams, they're getting imaged. And we asked, was there a correlation between pro-inflammatory cytokine levels and cognitive trajectory in these cognitively normal older adults? We hypothesized, of course, that pro-inflammatory cytokines would be detrimental, and the results were the opposite. What we found was that higher levels of interferon gamma and interleukin 12 in the plasma actually predicted a better cognitive trajectory. If you look here, interferon gamma, the red line is for folks who had just normal amounts or low amounts of interferon gamma in the plasma, their cognitive trajectory over 6 years going downward, but those who had higher levels of interferon gamma did much better over 6 years for cognitive trajectory. And interleukin 12 depended on the presence of amyloid in the brain. If you had high levels of amyloid in your brain based on high PIB, and you had low interleukin 12, this was your cognitive trajectory downward. For those with high interleukin 12, they did much better. That's the solid red line here. So interleukin 12 was dependent on amyloid in the brain. Interferon gamma was regardless of amyloid in the brain. So what this suggested is having an enhanced peripheral immune response could be beneficial for the brain. Why? It's specifically interferon gamma and interleukin 12 that modulate this T cell macrophage axis, where T cells are modulating where the macrophages can go into an M1-like state that is anti-infection. You have to induce macrophages into an M1-like state. T cells help this, and T cells do this by releasing interferon gamma. Macrophages release interleukin 12 to get the T cells to produce the interferon gamma. So when that axis is ramped up, it seems that in the periphery, in the blood, that's better for the brain. So there are different hypotheses for why that might be so. One of them gets back to Rob Moyer's, my late colleague's hypothesis. Many of you know the great Rob Moyer tragically passed away a couple of years ago. And it's Rob who first proposed that A beta isn't just junk, it's an antimicrobial peptide, first finding that it hits 8 clinical pathogens, but mainly it was in these mouse models showing that high amounts of A beta and amyloid in the mouse brain protected the mouse from salmonella infection, from herpes infection. In the contrast, in APP knockout mice, salmonella infection led to faster cell death. So this was the original data from Rob, and this was also a big result from Rob's paper showing that in young Alzheimer's mice, 4-week-old 5XFAD mice, there's no amyloid in these mice yet, but if you inject salmonella into the brain, then you can see amyloid plaques within 48 hours, and you can see in yellow here the red fluorescent salmonella co-localizing with the plaque. In fact, one bacterium could induce a diffuse plaque within 48 hours. Heat-killed bacteria don't induce, and in these young mice, you otherwise don't have amyloid. So this was proof of concept that a microbe could rapidly nucleate and seed amyloid. Same thing with viruses like herpes virus, HSV1, HSV6. Here we did it in the dish model, also in mice. And we can see in the dish that if you put these viruses into our 3D model, instead of taking 4 weeks to get an amyloid plaque, you get it in 48 hours. So again, this is a microbe seeding and nucleating amyloid proof of concept studies. Now, in new unpublished data, what Will Eimer has found in our group is that herpes viruses with naive neurons in the 3D model, if you introduce herpes virus, that they rapidly induce tangles from endogenous tau, and you can actually see that phospho-tau is co-localizing with the herpes. Now, normally herpes spreads in the brain by traveling neurotropically down axons. So this is how viruses, they go down the axon, they jump over a synapse into the next neuron. So this led to the hypothesis that Will's been studying. Is it possible that tangles are induced by viruses and by A beta oligomers that can be seeded by viruses or other microbes to block the neurotropic spread? Could tangles act as a roadblock for the spread of herpes down axons? So to test this, he created, this is unpublished data, this microfluidic model. So you have three chambers, a left chamber, a middle chamber, and a right chamber. And they're connected by microfluidic, laser-etched microfluidic channels where axons can grow in these channels, okay? So what you can see is if you have naive neurons on the left and you introduce herpes virus in red into these neurons, and now in the middle, you can either have naive neurons or A beta producing neurons. And then on the right side, you have naive neurons again, and you're looking at the rate of herpes virus spreading across these three chambers. And what you can see is if you have neurons making A beta in the middle versus naive neurons, if you look here on the right, with naive neurons in the middle, this is how much herpes gets through from here to here. But if you have A beta producing neurons, you see less virus getting through. And if you have high A beta producing neurons in the middle, you hardly get any virus through. And it's in these high A beta neurons, if you look at the phospho-tau levels, that the A beta is inducing phospho-tau together with the herpes virus, and the tangle formation in the middle is blocking the spread of this virus. So this led to this modification, extension of Rob's antimicrobial protection hypothesis, that perhaps amyloid plaques and tangles, and obviously already we know neuroinflammation, evolved as an orchestrated innate immune response to provide host defense against brain infection. And along the way, the genes that predispose to higher amyloid or tangles or neuroinflammation, like the many GWAS hits that predispose to greater neuroinflammation, perhaps those mutations had selective pressure for conservation because during times of great epidemics and pandemics of infection, the people who could more regularly induce neuroinflammation or even amyloid plaques or tangles were protected. So the idea would be that, even though we don't have really good enough data now to say microbes are inducing Alzheimer's today, is it possible that despite that, the Alzheimer genes that were conserved in mutations that were conserved that predisposed to this pathology over tens of thousands of years to fight these microbes, now these gene mutations are driving AD today, but perhaps it was the defense against microbes that led to the conservation of these genes. So that's a new hypothesis that we are now thinking about. So now let me go on in the last part of my talk to talk about how we can use our model systems for early intervention. And again, these are the systems here. So let's start again with this diagram. And step one, blocking A beta production. We know that gamma secretase inhibitors were not safe, they failed. Beta secretase inhibitors also not safe, they failed. But 20 years ago, my late colleague, Steve Wagner, who just passed away this past year, tragically, he and I decided, hey, let's just learn from early onset FAD mutations and find compounds that reverse the ratio of A beta 42 to A beta 40. And we call these gamma secretase modulators. So after 20 years of development and going through thousands of compounds, we finally landed on a clinical candidate that very effectively reduces A beta 42, somewhat hits A beta 40, and also increases A beta 38, which has recently been shown to be protective when there are high levels of A beta 38 in the CSF. The way this works is basically these modulators open the mouth of gamma secretase, the docking site. The presenilin mutations close the docking site. The APP mutations, in most cases, make it tough for the CTF to fit into the docking site. So if you open the mouth, you basically make more of the short A beta than the longer A beta that happens when the mouth is closed. So that's what these modulators do. First, our gamma secretase modulator now is an IND development. We expect our gamma secretase modulator to be in phase 1 safety trials in the fall of this year. It's tragic that Steve Wagner won't be able to see these, especially since he has led this project in collaboration with us for the last 20 years. But I hope he's looking down and seeing how we do. The second step, can we block A beta oligomers from inducing tangles, right? So we know now from the 3D models that oligomers induce tangles and synaptic degradation. So for this, we did a high content confocal screen with Steve Wang, and we screened 2,640 approved drugs and other drugs that have been through phase 1. And we also have been screening natural products in this screen. And what we found was 38 different drugs in this library, plus 43 natural products that reduced the phospho-tau to tau ratio, reduced tangle formation in the 3D model. Interestingly, the vast majority of these didn't do so by hitting A beta, A beta oligomers were forming in the dish, but these compounds blocked A beta oligomer from inducing phospho-tau entangles. This included GSK3 beta inhibitors, mTOR inhibitors like rapamycin was one of our hits, different CDK inhibitors, HDAC inhibitors, and AMPK activators, including metformin, which was a hit in this dish. Next, we can try to mimic Aduhelm. Aduhelm or aducanumab induces microglial clearance of A beta. Of course, Aduhelm is expensive, not particularly safe, but can we do the same thing with cheap drugs and natural products? And at the same time, we can ask, can we find drugs and natural products that prevent the pro-inflammatory cytokine release from activated microglia, which leads downstream to astrogliosis, inflammation, and cell death? So we did these two screens using various microglia, including BV2s and also IPS-derived microglia. And the bottom line was to find compounds that take pro-inflammatory activated microglia and turn them back into benign housekeepers that clear A beta and other debris by phagocytosis. This just shows some of the hits. Cat's claw extract here. This is a woody vine from the Amazonian rainforest that worked very well. This just shows resveratrol, quercetin, genistein as examples of natural products that reduce the release of pro-inflammatory cytokines like TNF-alpha, IL-6, following LPS induction of reactive microglia, or adding the microglia to our dish model. The final step is, can we try to find compounds that protect neurons from dying? All of this is going wrong, but can we protect neurons from dying? This was actually one of the first screens we did, and I just want to show you this result here. These were two compounds that were used in a combination. This was an idea from two students who came to see me 7 years ago from Brown University. The idea was to use sodium phenylbutyrate and a taurine analog to simultaneously prevent mitochondrial dysfunction inducing apoptosis, or stop ER stress and unfolded protein response from inducing apoptosis. That's what this combination drug does. The first trial we did was in ALS, and I wanted to state for disclosure, I am a founder of Amylyx, the company that did this. The ALS trial was published in New England Journal of Medicine, headed by Chief Merit Sokovich. And now this combination has been approved for ALS in Canada just over the past week. It's under priority review at the FDA. We'll hear by September whether it's approved for ALS from there. But we've also trying it in Alzheimer's disease. And in a phase 2 trial that we just released the results at CTAD, we saw that the same combination dramatically hit the A beta 42 to 40 ratio in CSF in the right direction. In other words, the 42 to 40 ratio went up, indicating less amyloid in the brain. Phospho-tau 181 significantly went down versus total tau. We also saw, not shown here, a reduction in neurofilament light chain, indicative of less neurodegeneration. So this phase 2 trial based on biomarkers was interesting, and now that's heading toward phase 3. So here we have then the total tally, 60 different drugs and natural products that induce uptake of A beta by microglia, or including the gamma secretase modulator that safely hits A beta production without blocking gamma secretase function on Notch and other substrates. 31 different drugs and natural products that prevent A beta oligomers from inducing tauopathy. This is the first drugs of this type. And then 68 different drugs, including approved drugs, natural products that prevent the release of pro-inflammatory cytokines from activated microglia that have been activated either by amyloid or by tangle pathology and neuritic dystrophy here. So now we're taking these compounds and we want to get them into biomarker-based clinical trials. And our mantra is right patient, right drug, right time, right trial, meaning these would be mainly small pilot biomarker endpoint trials matching the pathological biomarker to the pathology that particular drug or natural product targets, doing small pilot trials. These are being done at the McCann Center for Brain Health that I co-direct in collaboration with Stephen Arnold at Mass General. So this is the diagram, and as we find these various compounds, those that hit amyloid entangles and clearance of A beta we think would be most useful for early prevention, not so much for treatment of symptoms. Those that protect neurons from dying, like the Amylyx drugs, we think we can take a shot at symptoms. The big question will be, if you hit neuroinflammation, microglial activation, astrogliosis, will those be more proximal to being good for prevention, or will they also be able to treat symptoms of Alzheimer's disease? I think we'll start seeing some of the trial results in this regard over the next coming year or two. So I'd like to stop there and thank the Cure Alzheimer's Fund and NIA for my funding, also JPB, Good Ventures, Open Philanthropy. These are the professional staff and the Genetics Agent Research Unit who did this work, and my many collaborators around the world who help with these studies. Thank you very much for your attention.